So in the last section, we started introducing the idea of neurotransmitters and neuromodulators, and we're going to talk about specific specific chemicals in this section. And so there are lots of different neurotransmitters and neuromodulators, and you don't have to know all of them, but we're going to talk about a few of them that have a particular effect that we might run into in physical therapy. So we're kind of focusing it in a little bit more. So I want you don't have to know everything about them. You don't have to know anything about their chemical structure or the names of the receptors they bind to or anything like that. But I want you to be able to name them and briefly describe their effects and whether they're excitatory or inhibitory. So say a neurotransmitter, it can be excitatory, inhibitory, or both, depending, and sort of what its major functions are. So we'll discuss that in this lecture. I want you to know what determines the effect of a neurotransmitter. So a lot of these neurochemicals, they can act as neurotransmitters, they can act as neuromodulators, they can be excitatory, they can be inhibitory, and their effect changes depending on where they bind. So if they bind to a certain kind of receptor, they're going to have a certain effect. If they bind to a different kind of receptor, they're going to have a different effect. So the neurotransmitter itself doesn't determine what its function is. It's where it binds determines its effect. I want you to compare the relative speed and longevity of the effect of neurotransmitters and neuromodulators, and we talked about that in the last section. So neurotransmitters act fast and don't last as long. Neuromodulators act more slowly and last longer. So that's it. That's the relative speed and longevity in a nutshell. So I said that the effect of a neurotransmitter depends on where it binds. So a lot of times at the synapse, a neurotransmitter is going to bind to a ligand-gated ion channel. So ligand means bind. The neurotransmitter binds, it opens the ion channel, allowing ions to cross the membrane. So channels consist of proteins that function as receptors for the neurotransmitter, and they are those ionotropic receptors. So membrane channels are proteins. Neurotransmitters are largely proteins or opioidpeptins. So these are coded for by the DNA in our cells, in our neurons. So if you change the expression of different genes, you can change what types of receptors are there. And so keep that in mind in the next chapter when we start talking about neuroplasticity and how a neuron can change its chemical profile. So the ligand-gated ion channels, the gates open in response to a specific chemical ligand, which is the neurotransmitter in this case, binding to the receptor surface. And the gates are closed in its resting state. So basically it's that gated community that I was talking about. In its resting state, the gates are closed. You need to have a key, in other words, a neurotransmitter, to open the gate. The gates open rapidly and briefly when a neurotransmitter binds to the receptor side of the membrane channel. The ion channel receptors are like a lock and key. The protein membrane is the lock, or the receptor side is the lock, and the neurotransmitter is the key. And when you put the key in the lock, you open up the gate. Pretty simple analogy and very apt. Sometimes analogies don't go all that far, but this one goes really far. It has legs. So the G protein activation of ion channels change the electrical excitability of neurons. So there's a ton of information on G protein systems in the book, and I am not going to ask you a lot of things about G proteins, but just know that they have a wider spread effect than a single ion channel. So when a neurotransmitter binds to a G protein receptor, the receptor protein changes shape and the G protein inside the neuron becomes activated. It breaks free and it moves around and does stuff in the cell. So it can activate ion channels. It can activate gene expression changing the profile, the chemical profile of the neuron. So G protein receptors are a very powerful system for making changes in our neurochemistry. But that having been said, I don't expect you to know a ton about them. When you look at the learning objectives for this module, there's nothing about G proteins, but I just want you to know about that, know that they exist, and that's it, and that they do more than just a plain old ligand-gated ion channel. So a lot of the drugs that we use for treating different things are neurotransmitter agonists or antagonists. So agonists are drugs that bind to the receptor and mimic the effects of naturally occurring neurotransmitters. So L-DOPA, or Sinemet is the brand name, is a drug that mimics dopamine in the brain and takes and mimics the effects of the naturally occurring dopamine that is not being made by your brain when you have Parkinson's. The opioid drugs are drugs that bind to opioid receptors in your brain and mimic the effects of our endogenous opioids. So those are neurotransmitter agonists. Antagonists are drugs that prevent the release of neurotransmitter or bind to the receptor and impede the effects of a naturally occurring neurotransmitter. So there are those drugs like tramadol and some of the other ones that bind to the mu opioid receptors and prevent the binding of either naturally occurring opioids or opioid drugs. And so they block the effects of those. There are a lot of other drugs that are antagonists. So an antagonist slows everything down, and an agonist mimics the effect of a naturally occurring neurotransmitter. So it's interesting that we can use those receptor sites in our nervous system for treatment. So just talking about the specific neurotransmitters and neuromodulators. So remember, a lot of these chemicals can act as either neurotransmitters and neuromodulators depending on where they're released. And I don't expect you to know the details about them. I don't expect you to know their chemical structure or anything like that. Just generally what they do and whether they're excitatory or inhibitory or both. So glutamate, glycine, and GABA, the three Gs are the main neurotransmitters and neuromodulators in the central nervous system. They're all amino acids, so they're all building blocks of protein. They're all coded for in DNA. Glutamate is the principal fast excitatory transmitter of the central nervous system. It elicits neural changes that occur with learning and development. So it's a really important neurotransmitter in our brain. There are effects that we'll talk about in Chapter 7 of excess glutamate causing cell damage. So it's one of those things where some of it is good and too much of it is not good. Glycine and GABA are both inhibitory neurotransmitters in the central nervous system. Glycine inhibits postsynaptic membranes, primarily in the brainstem and spinal cord. And GABA activates slow-acting responses that are linked to ion channels via second messenger systems. So you can think of the three Gs, glutamate, glycine, and GABA. Glutamate's excitatory and glycine and GABA are inhibitory. And they're all amino acids. Acetylcholine is the major conveyor of information in the peripheral nervous system. It's used by all neurons that synapse with muscle fibers. And when you have that neuromuscular junction, acetylcholine is always excitatory at the neuromuscular junction because those are the membrane channels that exist there, those excitatory membrane channels. Acetylcholine also acts as a neuromodulator in the sympathetic and parasympathetic nervous system, in the autonomic nervous system, regulating heart rate and other autonomic functions. It's usually an excitatory neuromodulator. And receptors that bind acetylcholine are responsible for a wide range of effects in the nervous system. In the central nervous system, acetylcholine functions in the selection of objects of attention. So acetylcholine is all over the place. The place where we encounter it the most in the peripheral nervous system is at the neuromuscular junction. But it also is one of the major neurotransmitters of the autonomic nervous system. So just think about it as it's primarily an excitatory neurotransmitter or neuromodulator, and it's all over the place. So this group of neurotransmitters and neuromodulators are called amines based on their chemical structure. You don't need to know that. Dopamine, norepinephrine, and serotonin. And histamines, even though histamine acts as a neuromodulator in the system, we've talked about it with regard to allergic reactions and cellular reactions to injury. So I just put it on this list because it is an amine like the other ones, but the main ones that I want you to know are dopamine, norepinephrine, and serotonin. Dopamine has, again, wide-ranging effects. It can be either excitatory or inhibitory depending on the receptor. It affects motor activity, cognition, and behavior. So that's a wide-ranging group of effects right there. Norepinephrine plays a vital role in active surveillance of our environment by increasing attention to sensory information. It can be either excitatory or inhibitory depending on the receptor, again. So norepinephrine is a heavy hitter in the autonomic nervous system in our fight or flight response, but it also has other effects throughout the nervous system. Serotonin affects sleep, general arousal level, cognition, perception, motor activity, and mood. It's usually inhibitory. So its receptors just happen to be ones that are inhibitory. You hear serotonin with regard to mood. The drugs that are generally considered antidepressant drugs are selective serotonin reuptake inhibitors, so SSRIs. So they actually allow the serotonin to stay longer in the synaptic cleft to modulate our mood, which is kind of neat. So and histamine is concentrated in the hypothalamus, so it's released when it gets the chemical signal from cells in the body that something's going on, that we need some histamine down there. So it's more of a neuromodulator. I just put it on the list because it's the same group as dopamine, norepinephrine, and serotonin. So this is a graphic from the text, and I just love it. Well, you know, I like Venn diagrams, but you can see sort of the interaction of norepinephrine, serotonin, and dopamine on emotion, cognition, and mood. So all of these affect all of those things. So, and some of the effects, especially sort of what we consider to be negative effects like anxiety and irritability and aggression and harm avoidance, are interactions of two different ones. But where they all interact is they all affect emotion, cognition, and mood. So in the module, there's a link to a website that's called Mouse Party. And it's really interesting. It's from the University of Utah Medical School, I think. And I came across it years ago, and it's still up. And it's a little animated thing where it shows the effects of different drugs on the brain of a mouse and how it affects neurotransmitters. So there's a little graphic in there. You go in and you select a mouse, and it picks the little mouse up and puts it in the chair, and then it has a short little blurb on what's going on in the mouse's brain. So it's pretty cool, and it kind of shows you some of those effects of neurotransmitters. So a lot of neurotransmitters have effects that motivate us to do certain things, so that the motivation, reward seeking, hunger, that sort of thing, all associated with dopamine. And different drugs can have different effects on these neurotransmitters. So very interesting area of study. There are another group of neurotransmitters that are peptides. Opioid peptides, our natural endogenous opioids, they're endorphins, enkephalins, and dynorphins. They inhibit neurons in the central nervous system that are involved in the perception of pain. So those endogenous opioids are usually inhibitory, as are the drugs that mimic them. Substance P is another peptide that stimulates nerve endings at the site of injury, and those are usually excitatory, which increases the perception of pain. So it's the opposite almost. And calcitonin gene-related peptides act as neuromodulators, and they're excitatory or inhibitory depending on their receptor. Nitric oxide is the only neurotransmitter / neuromodulator that can cross the cell membrane without having a membrane channel. It doesn't require a receptor. It can just diffuse across the membrane. It regulates the vascular system in the periphery, and it's also active in the brain. So they've found that nitric oxide is involved in persistent changes in the postsynaptic response to repeated stimuli and in the cell death of neurons. So we'll talk about in Chapter 7 our response to repeated stimuli. And nitric oxide is one of the neuromodulators that affects that. So again, it can be excitatory or inhibitory depending on where it is. So synaptic receptors, which are the membrane channels, are typically named for the neurotransmitter or neuromodulator that binds to it. So the precise action of a neurotransmitter on the postsynaptic cell is dependent on the property of the receptors. That makes sense, right? So they're either going to be those direct receptors, the ion channels, which make up a single functional fast-acting unit, or the indirect actors, which usually cause a cascade of intracellular effects. And they cause changes within the postsynaptic neuron. So those ones are slower and they're longer acting. Postsynaptic receptors use two mechanisms to transduce signals. And this information is sort of repeated a couple times just so you can get it and hear it in a different way, because it's important to remember this. So either those fast responding ligand-gated ion channels or the slow responded G protein activation. So usually in most of the chapters we're going to talk about at least one disorder, so to sort of ground it clinically. And in the book it lists lots of different disorders, and I'm going to try to stick with the ones that you might see in physical therapy. So myasthenia gravis affects the neuromuscular junction. It's an autoimmune disease, as are a lot of the neuromuscular degenerative diseases. In myasthenia gravis, the antibodies attack and destroy the acetylcholine receptors on muscle cells. So with myasthenia gravis, it obviously affects strength and endurance. And I thought this was interesting, the epidemiological aspect of it. Myasthenia gravis in women typically occurs between the ages of 20 and 30 years. But in men, it's much later, in 60 and 70 years. And I don't think it's known why, but I think that's really interesting. I've worked with 4 people with myasthenia gravis, 2 men and 2 women. And one of them was a younger woman, one of them was an older woman, and then both of the men were older. Usually, myasthenia gravis is treated but with drugs that inhibit the breakdown of acetylcholine to improve function because they increase the amount of time that acetylcholine is available to bind with the remaining receptors. So they're sort of like, I talked about the selective serotonin reuptake inhibitors. They're sort of like acetylcholine reuptake inhibitors. So it just causes the acetylcholine to stay longer in the synaptic cleft. And so it's available to bind with the remaining receptors. So people who have myasthenia gravis, the receptor sites fill up and then they can't excite a muscle contraction. So in muscles that are used over and over again, like postural muscles and eye muscles, people blinking. And so a lot of times people have trouble holding their head up. They need to work on energy conservation. They need to work on strengthening, but at a very low level. So usually the 4 people that I've worked with myasthenia gravis, one of them was in a skilled nursing facility, and 3 of them were an outpatient. So usually they're being treated with the medications that are going to improve their function, and a lot of times we're working on strengthening, endurance, and energy conservation in physical therapy.